Because we will now finally be talking about different cytotoxicity evaluation approaches and what will happen and what can happen and what you should aim to obtain some proper results and so on. So lecture will be probably a bit longer than usual, and do not forget, in the middle we will have a test, maybe in 40 minutes or something like this. Well, and do not forget that after this lecture we'll have a presentation. The last, probably today not 3, but 4 people today should present their lectures. So 10 minutes after the lecture I'm waiting for you in Zoom room. And the last lecture we will discuss maybe in the end of this lecture. So let's proceed, do not waste some time. First one, we need again to discuss something about the concept of cytotoxicity, because when we are assessing some toxicity, it definitely can be a cytotoxicity. So cytotoxicity, it's toxicity concerning some cells, so damage done to cells. And cytotoxicity usually associated with in vitro tests, because, well, it's quite hard to assess some cytotoxicity when we are talking about a whole organism. And with the concept of cytotoxicity connected to terms, it's well, drugs, but it also can be just a substance. It's a cytotoxicity and not cytostat. I don't know even this word exists. So it's cytotoxic drugs and cytostatic drugs. Basically, if you're going to check some literature, you will probably didn't see any cytostatic drugs in comparison with cytotoxic. So most of the drugs are cytotoxic drugs, and in some sources I even see that the term cytostatic used only in post-Soviet countries. As you can see, cytotoxic drugs, they killing cells by necrosis, and we will discuss on the next slide that this is not quite correct. And cytostatic drugs looks a bit more, let's say, mild, not so harsh, because they're using to induce some apoptosis process in cells. But to be honest, now not many scientists using cytotoxic or cytostatic terms. It's just anti-cancer, maybe anti-tumor or something like this one, because it's really hard to distinguish what is causing necrosis or apoptosis or some other kind of cell death. So basically, if you are reading some article, maybe not from a very good journal, not from a Nature, not from a cell journal, you will probably see just 2 types of cell death. It's apoptosis, or regulated process of programmed cell death. No, not even programmed cell death, just a regulated process. And necrosis, it's a pathological process. Just remember this word, pathological process. And no unregulated, non-regulated process, and so on. If you're talking about morphological changes, there are different changes when we're talking about apoptosis, because cells which undergo apoptosis, they will shrink, so decrease in size, and there will be a lot of apoptosis bodies around these cells, so very small vesicles. And when we are talking about necrosis, it's definitely enlargement of a cell, so the cell will swell and increase in size, like exploding, and a lot of cell organelles will release in surroundings, and this causes inflammation. So but the problem is that already a lot of years ago, a very big group of scientists, as you can see here, it's like it's not a whole list. The whole list of scientists occupy maybe a couple or 3 of list of this paper, and there's more than 100 of affiliations. So it's a group of scientists that provide a new recommendation for different type of cell death. So for now, it can be 2 types of cell death. It's a regulated cell death and accidental cell death. And in regulated cell death, there's subdivision of programmed cell death. So if you are ever aimed to publish any paper in a very good journal connected with some cell cycling, so like Cell maybe, or some other like Nature, maybe Nature, not biotechnology, but Nature journals connected with biology. So in a very good journal, you need to know what is the real classification of cell death types. So what is so special about regulated cell death and programmed cell death? Even necrosis, it's a regulated cell death. What is accidental cell death? Well, if you are burned or you spill some acid on your skin, for example, and some of the cells dissolved in acid, well, it's an accidental cell death. All of other cell types, it's a regulated cell death in any ways. And what is the classification of the cell death when we are talking about different levels of different compounds? So one of the compound is ATP, and the second compound is reactive oxygen species. And well, there's some, let's say, some stages of cell death. So it's adaptive response. When you have increase in ROS in your cells, this increase can be like a sinusoid, and it's adaptive phase. But after some adaptations, there can be regulated cell death initiation when the ROS level is increasing and ATP is decreasing. And there's some point, let me just switch this off and switch this on. In case of there's also some point here. It's called a point of no return, and after this point of no return, it's execution of regulated cell death. So before this point, cell death and the different processes of cell death can be of necrosis or apoptosis behavior. So it's a lot of, sorry, I just need to answer because this wouldn't stop. Sorry. So it's just cell death can behave some features of both necrosis and apoptosis. So what I want to say is that well, there's not only necrosis and apoptosis types of cell death. There can be a lot of, for example, on different stages, cell dying because of apoptosis, let's say, can feature some processes looking like a necrosis maybe. Or necrosis can feature some process of apoptosis and so on. So it's very complicated thing and very complicated process. But what's next? This was released in 2015, and I just checked. There's another new recommendation of Nomenclature Committee of Cell Death a couple years ago. And as you can see, there's a lot of different types of cell death. All of this is types of cell death. So it can be necroptosis, ferroptosis, we discussed this, so peroptosis, parnatosis, and a lot of other things to mention. What I want to give you some additional information about this. As you can see, it's a very big group of scientists. And again, when you are talking about, when you are reading some papers in not very good journal, you will not probably see any of these cell death types. But still, if you aim to publish in a good journal, you need to know that something of this exists. And by the way, it's a very nice thing that I found during preparing to this lecture, is that just mention that the first author here and the first author here is the same man, just for you to, I don't know, to know what to aim for, because here's this man, and as you can see, he's very young and just 40 years, and just look at age index of this how it's Lorenzo Galuzzi. So he's the youngest and most cited biologist in Europe, and one of the most and one of the best specialists in cell death types and so on. Let's proceed. Let's stop with this apoptosis, necrosis, and so on, and just let's go to the cells and what can happen to the cells. So as you can see, there's a lot of cell types. So it can be different cancer cells, it can be epithelial cells, it can be different brain tumors, mice cells, human cancer cells, it can be different algas, unicellular algas. It can be even some insect cells, it can be plant cells, and so on. And most of the cells can be used, and there's an enormous amount of different cell lines in nature now. So you can use all of the cells, but the responses of the cells to different toxicants in some of the cases are very similar. So different necrosis and apoptosis process can undergo almost the same way in, I don't know, plants cultures or algae cultures, and in human cancer cell cultures, and so on. But anyway, there's a lot of processes that can happen to you already maybe see this picture. It's a very famous picture when we're talking about what can happen to nanoparticles after interaction, to cell when after interaction with nanoparticles. It's a lot of things can happen, and since a lot of things can happen, you need some instruments to assess this damage. So in case of cell membrane damage, in case of interaction of nanoparticles with mitochondria, in case nanoparticles damage some DNA or RNA, you need a lot of instruments, a lot of approaches to understand what really happened to your cell when when some nanoparticles accumulate inside. So there really can be a lot of approaches to assess this damage, and so we will talk about what you can use to assess this damage in your cells. First one, you always can use some microscopy. So microscopy, it's a basic method, and in most of the cases you need microscopy to observe something. And I just want to show you some video which showing a macrophage. We're talking a lot about macrophages, but maybe not all of you are familiar how the macrophage really looks like. So I will show you a macrophage which already eat some gold nanoparticles, and well, after eating the nanoparticles, this macrophage continue to eat some dead cell. So here it is. As you can see, it's a macrophage here, another macrophage here. They're looking for some nanoparticles, gold nanoparticles. As you can see, it's a dark field option for a microscope, and this is a dead cell, and so this macrophage trying to eat some of the dead cells and already eat some gold nanoparticles, as you can see here inside of macrophage. So you can use some like this one. It's a dark field plus maybe phase contrast or differential interference contrast options for a microscope, but still. But using microscopy, you can also observe some other options of the cell, and the first option is morphology. So morphology is a direct way to assess cell viability. When we are talking about morphology, there's a lot of cell morphology types, as we mentioned on previous lecture. They can be fibroblast-like, epithelial cells-like, and so on, but most of the cells, they are like spindle-like shapes, so have a very wide center, thin ends, and so on. And when the cell biologist or someone who working with the cells, working with them for quite a long time, it's already obvious when you're looking on cells. It's obvious what cell viability is, at least. Well, maybe plus minus some percent, but anyway, when the cells are 100% viability, you look on this monolayer and you are sure that these cells are alive. When you are looking at cell monolayer and something happened to cells, for example, some of them looks like a spherical, so they lost some connection with the tissue substrate, cell substrate, it's probably some damage to cytoskeleton, some decrease in viability, and it can be like a 70%. But still 70%, it's not a big decrease, it's just some decrease. But when you're looking on cells and you see some cells that expanded like 3 times, it's definitely some necrosis. And as you can see, there's a lot of some part of the cells. It's not a cells. You will probably not see a single cell, maybe this one, a single cell, maybe this one. But it's a lot of cell debris, cell shadows, how it's called. So it's just a cell membrane without any internal organelles. This is how cell culture looks like at a viability of just 40%. So 40% is still relatively big looking on this picture because there's no cells on this picture. No, but we will talking how this connect with each other, because this amount of live cells, like 40% of cells are alive, 60 are dead, but this is a result from indirect way. So again, this is direct way to observe. You observe it with your own eyes. And with microscopy you can observe like total amount of cells, different morphological changes like size and form maybe, and amount of adherent cells. For adherent cell cultures, it's very crucial that all of the cells should be adherent to a culture plate. But that's just your eyes. It's very hard to calculate the cells, to count the cells by your own hands, let's say, and that's why there's another options called flow cytometry, and you can use flow cytometry to count your cells using some flow cytometer, and we have this device in our lab. So flow cytometer can separate cells using different parameters. For sure, you can stain your cells with different dyes. We will discuss in next slides what you can use for this. But even without any staining, you can distinguish your cells by 2 parameters. It's front scattering and side scattering. So your cells, when they are passing flow cytometer, it's very narrow capillaries, and in this capillaries there's a light, and different cells, if you are talking about internal structure, so it can be a granulation of cytoplasm. The cells may shrink or may expand, so any of morphological changes will be represented on these plots. So either side scattering or front scattering will change, and based on these changes you will observe what happened to your cells. So probably in some of the previous courses you should have some additional information of flow cytometry, and of course you can plot some distributions and observe how many cells in different parts of this plot, how many cells undergo apoptosis, necrosis, and so on. But all of this is good, but these things based only on morphological changes mainly, and one of the most powerful instruments to assess the cell viability, it's fluorescence. So fluorescence, again, in most of the cases, is direct way to assess cell viability. Why direct? Because again, you can observe this fluorescence by your own eyes, and you can well directly understand what happened to your cells based on what you have stained and what and how and so on. If some of you not familiar with basic principle of fluorescence, it's very easy. There can be some in some fluorescent molecule there can be some energy levels, and if you have some photon with some energy, it can let's say energize the system from one transform system from one energy level to another energy level, and there can be non-radiative transitions and radiative transitions, which result in emitting of additional photons. And when we are talking about common, let's say, fluorescent dyes, usually it's just a Stokes shift. So Stokes shift, it's a process. For example, if we have some absorption of the light, the emitted light will be of longer wavelengths, because the longer wavelengths, the lower the energy of this light, of this photon. So because of non-radiative transition, some of the energy lost for heating, basically. And so the Stokes shift means that if you have illuminate your cells with blue, the emission will be maybe red or maybe yellow. But there's also anti-Stokes shift, and on this principle work some upconversional nanoparticles using some radiodopterous elements and so on. So in anti-Stokes shift you can observe opposite process. When you have some near-infrared light, you illuminate your particle, and your particle will produce some visible light. So these upconversional nanoparticles are widely used when you need to, let's say, transfer some energy inside a human or animal organism, because animal organism it's penetratable for near-infrared light, not for visible light. And for example, you can illuminate your tissue, your skin with infrared light, and then nanoparticles will convert this to visible light, and this visible light can next illuminate some, for example, dyes in a visible area and produce some photosensibilization effect or something like this one. But that's just the basic principles. And fluorescent staining, it's not very easy after all, because you need to answer a lot of questions. And these questions usually connected not only with the person who will perform fluorescence, but also with you who will ask, stain me something, I want to, I don't know, see something, what happened with nanoparticles in the cells, what happened to cells after interaction with nanoparticles, and so on. So everyone should understand what you want to obtain from stained cultures. So the goals can be very different, because the first question, of course, what do you want to stain? Because considering current advances in biological and chemical sciences, there's currently almost every, maybe not even almost every structure in cell can be stained. And starting from nucleus and cell membranes, all of the organelles membranes and lysosomes, and I'm sure that even peroxisomes and so on can be stained. So not just stained, but differential staining. On next slide we will discuss this. So as you can see, for example, on this picture, it's a whole process of mitosis, and as you can see, it's a chromosomes, it's a tubulin, it's centrioles in cytoskeleton, everything is stained here, and on combined image you can see a whole process of mitosis. So it is possible. You just need to pick up a correct dye. So it's with blue excitation, it's with green, it's with red. So it's three main RGB colors. And so the next question is, do we want to see the process? Because here we see a process, but this is just an image, and this is probably an image of a fixed cell. But you can also see some process, for example, of endocytosis. So how the cell uptake your particles. You can observe this in real time with some, for example, lipophilic staining of membrane. So you will see how this pseudopod is forming and how they uptake your nanoparticles and incorporate these nanoparticles inside the cytoplasm. So even process visualization is available now. And what kind of sample quality do we need? So, and the next question, will we observe the sample again? Because it's an important question. Do we need to fix a cell with different fixed solutions? Do we want to observe the sample again? So do we need to seal it, to store it, I don't know, seal with some preservation agents and so on? It's a question about quality of the sample. So a very good glasses, a very good cover slips, and so on. Now, or we just need to observe this in routine practice. So just observe and throw it to a trash. And this is important questions after all. And about staining process, it's also a lot of questions because what kind of fixation should we apply? So if we need to observe the sample again, we should fix our cells with formaldehyde or something like this on. And what kind of fixation should we use? Will this fixation interact with nanoparticles, with cells, and how? We need to understand. What is the concentration of the dye? Because overstaining or understaining is definitely will produce, especially overstaining, will produce some false positive results or false negative. What is the time of staining and what is the time and concentration? Well, it's some things that should be picked up empirically by a specialist who performs the staining. But still, it's a good questions. And how to interpret the results. So when you stain something, you need precisely know what you are staining and how it should looks like. Because if you are using some dyes and you do not know what should it be stained and so on, it's very complicated to understand what you obtain after all. Well, probably again we will discuss it a bit later. So what about fluorescent staining? Fluorescent staining, in other words, also can be called a differential staining. Why differential? Because you need to differentiate something. So basically you never stain your cells just to stain. You need to distinguish, for example, live cells from dead cells, or one organelle from another organelle, or one cell type from another cell type. So you need to differentiate something from something, find some difference. That's why fluorescent staining called differential staining. And for example, in these pictures, you see some of the examples of very frequently used staining techniques. The first one is FITS label tennexine plus prapidium iodide. So in all of these examples, you see prapidium iodide, as prapidium iodide stains the nucleus of dead cells in red. So as you can see, red here, well, nothing red here because all of the cells is alive. But still, prapidium iodide very frequently used since, I don't know, 17th years of previous century. And why of the dead cells? Because prapidium iodide cannot penetrate live membrane. So there is no way for prapidium iodide to penetrate the membrane of the live cells. So if the cell is stained by prapidium iodide, it's just not a definitely dead, it's already undergoes some processes of destruction. So the membrane is badly damaged. And what's so special about this first one, annexine labeled FITS, is that this dye can be used to stain cells in 3 different conditions. When you have just a green fluorescence, it means that some annexine binds to phosphatidylserine on the outer cell of the membrane of dying cells. So if your cells are just green, it's a first step of cell death. If some of the cells, like here, green and red, it's some kind of middle process. So they already have some permeability of membranes, but still they're in the middle of the cell death cycle. And if you have some cells only with a red nucleus without any of the green fluorescence, it's the last stage of cell death, or maybe it's just a necrosis type. Necrosis death, let's say death with the features of necrosis, to be correct. So using just one kit, usually this provided as a kit, so you just use one kit, you will obtain information on 3 let's say 3 cell populations concerning death cycle. Ecdin orange also very frequently used to stain some cells, and what's so special about Ecdin orange as it can be used to for differential staining, because it can stain DNA in green, and as you can see there's some DNA in, especially in nucleus of living cells, and single-stranded DNA which can be found in dying cells. So upon destruction of nucleus, different restrictase and so on, they disintegrate of DNA to a single-stranded DNA and so on. So single-stranded DNA and RNA, as they are single-stranded, they can be stained in orange. So ribosomes will be probably stained in orange because there's a lot of RNA in ribosomes. And also propidium iodide stains the nucleus of dead cells in red. So green cells with red nucleus is dead cells, just green cells it's living cells. And fluorescent diacetate plus propidium iodide, it's a common dye for flow cytometry. So living cells again FDA plus, they are green, and dead cells they are propidium iodide positive, so they are dead cells. As you can see here, only living cells shown. And so it's just what can be stained, basically a live dead stain as it's called, but we're talking a lot about reactive oxygen species. And as you might understand, there is no way to stain molecules, separate molecules, especially not even the molecules, but some ions and so on. So how the ROS can be stained? Well, there is of course some options to stain some not ROS, but to detect their presence in cells. So the first one, the first staining is called DCFDA, or sometimes it's called dehydro-DCFDA. It's a staining which used to detect some hydrogen peroxide related reactive oxygen species. For example, if you're using just as we discussed on previous slide, just the FDA, fluorescent diacetate, in living cells, as you can see, it's control cells. Living cells will be stained in green. When you treat your cells with hydrogen peroxide, all of the cells will die, and you will observe very shallow color, green color. But if you have some inhibition of hydrogen peroxide, you will again observe some green color again in your cells. The opposite situation when you are using the CFDA. So when you're using the CFDA, in control cells, you should see nothing. So almost nothing, because there is no additional reactive oxygen species. But if you add some hydrogen peroxide as positive control, you will see cells. So just not to, let's say, don't mess FDA and CFDA, because they are resulting in completely different staining patterns. And how it's working after all. This is a picture from one of our articles with magnetite nanocontainers. As you can see here, all of the black dots on fluorescent images, usually it's nanoparticles. Because they are not quenching fluorescence, they are just quenching the light, because, well, they are impenetrable. And how the CFDA works? So on the first stage, after penetrating the cell membrane, some cellular esterases they already cut some of the part, so diacetate part of this dye, and reactive oxygen species, especially here in the process of Fenton reaction, they are cutting again this hydrogen addition, and in the result you obtain some fluorescent diacetate, which is fluorescent. So if you have no reactive oxygen species, there will be no fluorescence. As you can see, in control samples there is no fluorescence. in sample incubated with magnetite nanocontainers, well, again, a couple of cells maybe. It's a low concentration of hydrogen peroxide, and this is a high concentration of hydrogen peroxide. So as you can see, the result is obvious, no reactive oxygen species generation here, and well, positive control shows a lot of ROS accumulation in cells. But there also can be some other approaches to stain something connected with ROS. For example, it's a body pie staining. Well, body pie, it's a whole family of different molecules, which is used to stain different kind of things happen to a cell, but this one is body pie staining for lipid peroxidation. So lipid peroxidation, it's again one of the main processes of cell damage by reactive oxygen species, and well, it's called here, as you can see, trademark body pie. Body pie with this exactly wavelengths of excitation and emission, usually excitation and emission for body pie in a very narrow region, as you can see, just a 10 nanometers difference. But how it looks like, for example, it's cells which were treated with acetylcysteine, as I can remember correctly, and untreated cells and treated cells. For example, untreated and treated with some reactive oxygen species generation compound, and treated again with antioxidant compounds. So here is untreated, here is treated. As you can see on the middle pictures, it's a lipid, let's say lipids, which undergoes peroxidation. And on a merged image, you can see some difference. So just for you to know, yes, it's an option to stain some lipids, peroxide lipids in your cells. Well, also it's an option for flow cytometry to use this dye in flow cytometry, and for example untreated and treated, it's very easy to understand is it works or not. And well, it's about staining of reactive oxygen species. Of course, it's a lot of other options, but still you can learn it by yourself. Co-localization. When we are talking about probably after this couple of slides, we will have a test. Be prepared. So co-localization. What does this mean? It means that you need to understand in some cases where is your nanoparticles accumulated in your cell, and this can be important to because this will probably provide you information about damage. So some of the very first information about damage. Probably here, it's a co-localization of gold nanorods with, well, some cell organelles, which is stained by rhodamine 123. As you can see, probably not very good, but still. It's a staining of cells with rhodamine plus propidium iodide and rhodamine staining mitochondria. So it used not to stain mitochondria, but to observe a mitochondria potential. And according to this potential, this fluorescent dye can fluorescent differently. And on the second picture here, it's a dark field image of, well, dark field, it's a let's say option to observe a side scattering of your nanoparticles. So you have a nanoparticle, you have a light coming from the side of this nanoparticle, and you observe the light scatter to 90 degrees into your objective of your microscope. So it's an option to observe nanoparticles using optical microscope. As you can see, there is no way to observe something that is smaller than half of the light wavelengths, but in dark field you can observe by scattering. So if you merge these images, fluorescent images and dark field images, you will observe some green looking areas. As you can see here, it's a very pale but still bluish greenish color, and this is the mitochondria. And as you can see, there's a lot of nanorods can be observed co-localized with this staining of mitochondria. And you can probably, I mentioned this specifically, just probably, that some of these gold nanoparticles can damage mitochondria, and this can cause a cell death. But again, it's just a first step. So for you just to understand what can happen in first assumption, and what else you can observe. For example, here, it's a different staining of the cells after accumulation with silver nanoparticles, silver nanospheres, and in some cases it's impossible to understand where this nanoparticles is located. For example, here, just a common FDA staining, and as you can see, yes, there's some nanoparticles, definitely nanoparticles, aggregates of nanoparticles, but since FDA stain everything in the cell, it's a cytoplasm staining, it's a nucleus staining, everything is stained, you can distinguish anything inside the cell, and the whole thickness of the cell is stained, so the stain is everywhere on every stage. So it's probably impossible to understand where is the nanoparticles. And if we are looking on acridine orange, it's another situation. Why? Because you here can see some intercellular organelles, like a micronucleus, a nucleus, some ribosomes, as I mentioned, they stained in orange, and you can, because acridine orange do not stain cytoplasm, because there's almost no DNA inside cytoplasm, you can observe the cells layer by layer, as well as in confocal microscopy. So again, on next slide will be. And if we compare, and well, NXN fits gives almost the same picture as FDA, because here you can definitely understand that this just adjusting focus of your microscope, you will definitely see that these nanoparticles located in the middle of the cell. So where is nucleus, for example, located? And here you cannot see this because you see no nucleus, you see no membranes, you even in some cases you see no difference between cells. So it's just a lot of staining of everything inside the cell. So when we are talking about co-localization, it's important to pick up a right dye for cell staining. But what about confocal microscopy? Well, I wouldn't explain your basic principles of confocal microscopy because it's not my duty, but still confocal microscopy allows you to focus on a specific focal plane in your sample. On the focal plane, I mean that you can optically slice your cell on different, well, slices. And here's an example. It's a slices separated by 1 micrometers. So very good confocal microscopes can slice even on, let's say, smaller slices, like maybe 300 nanometers, 400 nanometers, depends on laser wavelengths. And slicing the cell, you can definitely reconstruct the cell to a 3D picture. You can observe some processes with confocal microscopy. For example, here it's a stained lysosomes. And more important, you can slice your cell in the middle, and if you observe some nanoparticles in this middle section, you can 100% be sure that these nanoparticles inside your cell, not outside, underneath the cell, and so on. So confocal microscopy, it's a main instrument for you to understand if something inside the cell or outside the cell. So for co-localization. And there's just some middle section of nanoparticles. For example, this is a cell, it's a very big vacuole, and in this vacuole you can see some gold nanoparticles. So maybe not a vacuole, maybe some big endosome. So, fluorescent staining related problems. Definitely there can be some problems with fluorescent staining, and the first one, since we're talking about nanoparticles, There can be some problems connected with interactions of fluorescent dyes with nanoparticles. So interaction of nanoparticles with fluorescent dye doesn't matter. Here just two examples, but of course there can be a lot of different examples, but this is just from my practice. There's two types of nanoparticles. It's a gold nanostars, it's a gold nanospheres with a diameter of 50 nanometers. So they're both like 50 nanometers. And what about staining? For example, it's a DAPI staining. So DAPI staining used to stain nucleus of the cells, and DAPI cannot penetrate living cells membrane. This is prapidium iodide. Again, prapidium iodide cannot penetrate living cells. And this is acadine orange. What so special about these pictures? As you can see, after incubation with gold nanoparticles, which are in most of the cases are not toxic, you can see just a couple of cells are blue, so a couple of cells are dead. Same here, just a couple of cells are red, so they are dead, and you can see acadine orange, fluorescent, very nice, in green color, so everything is good. But what happened when you are using gold nanostars? So why every cell are stained like a dead, and you can see not a green but some yellowish color? And well, that's what was not so obvious when we obtained these results, because well, no one ever thinks that some of the precursors may be left in media in these nanoparticles. So as I mentioned on second lecture about the synthesis, there are some surfactants that are very frequently used to obtain some morphologically complex structures like gold nanostars. So in this case, there was a STAP stabilized gold nanostars, and gold nanoparticles, they're just electrostatically stabilized without any coating. And it was found that the concentration of STAP, which is very toxic, in the case of these gold nanostars, the concentration of STAP is so low that this concentration can't cause any toxicity, cytotoxicity, but it's enough for staining to penetrate the cells. So in all of these cases, as you can see, in all of these cases, all of these cells, they are alive. Maybe these particular cells, as you can see, they are much more bright in prapidium iodide, and maybe some of the cells, like maybe this one or this cells looks like dead cells. But still, this staining not because of the dead cells, the staining because of the membrane damage. It's the same as I talked to you about damaging of membranes in controlled manner when you want to alter their viability. So, but a special attention should be paid to this picture, because we see here not just enhanced staining like in case of these dyes, but we have changing in color. So why the color have changed from green to some yellowish to even some kind of brown color? Well, because as you probably remember, I told you that acadine orange can stain different type of organelles in different colors. And what again so special about acadine orange, there is not just DNA in green and RNA in orange or red. It's also acid. In acid condition it's red, and in normal condition it's green. So it's also difference in pH can be detected using acadine orange. And since it's a surfactant, it can damage endosomes and lysosomes. And this means that pH of inside the cytoplasm of the cell will decrease. And upon decreasing of pH, we will observe some additional red staining. So if we add some red to green, we will observe something like this one. So when we are using some nanoparticles, you should always understand what your nanoparticles consist of, and what are the stabilization agents, and how, in which manner they can interact with your nanoparticles and alter the results of your staining. Because sometimes you can obtain completely different and very strange results. And in case you're using some staining which is not standardized, let's say, so not just some kits from Helicon or something like this one, you need to properly understand where your new nanoparticles with some fluorescent behavior may accumulate. So in case of gold nanoclusters, for example, it's just a 20 aurum atoms stabilized by bovine serum albumin. In case of this staining, there was observed very different accumulation of, so they're fluorescent in red. And as you can see, there was observed a lot of accumulation inside of cytoplasm. So to understand what is fluorescent here in red and what is fluorescent here in blue, you need to understand how this gold nanocluster metabolized by cells. So in case they enter the cell, they still fluorescent in red, as they should be. So in red, probably, despite this one, because this was like maybe a couple of cells with a fluorescence of everything inside the cell, but it's a separate case. In most of the cases, there was definitely some blue fluorescent organelles and some red fluorescent organelles. And red fluorescent organelles, it's early endosomes or just endosomes. So when these clusters penetrate the cell membrane, they retain the ability to fluorescent with red. But after interaction with lysosomes, so in late endosomes or in endolysosomes, some of the bovine serum albumin get dissolved, restricted by enzymes, and so the gold nanocluster stop fluorescing, because it's important for this cluster to have both atoms and stabilization molecule. And the remaining fluorescence is just autofluorescence of BCA, and this autofluorescence are in blue. So the late endosomes or exosomes stained in blue because gold nanoclusters got dissolved, and the early endosomes or just entered endosomes, they're staining in red because the gold nanoclusters are intact. But in case these nanoparticles enter the cell during the stage of mitosis, it can happen very strange things, because as you probably know, during mitosis, the nucleus membrane, which is in rest of the time impenetrable by different dyes and so on, the nucleus membrane like dissolving, and at this stage, some of the nanoclusters can enter this cell and enter this nucleus and retain in this nucleus even after dividing of the nucleus. And during the mitosis stage, there is no endosomal transport, so no endosomal, no exosomal, so the cell just dividing and do not spend energy on other processes. So in this case, all of the nanoclusters which were accumulated in sorry. So in this case, all of the nanoclusters which were accumulated in cytoplasm retain in cytoplasm without being dissoluted by lysosomes. And so we see no blue-colored exosomes or blue-colored late endosomes in this. In this cytoplasm, only endosomes, or maybe not even endosomes, just some clusters in cytoplasm. So you need to precisely know what happened to your cells, on which stage your cells. It was, I mentioned in previous lecture, you need to understand what is the stage of your cell culture, because it can be synchronized cell cultures. you can found your cells all in a stage like interface and stage G0 when no of cells will divide or so on. So it's important for you to understand how the cells behave, to understand how they will accumulate, can accumulate, and so on. But also there's some problems with image processing. For now, it's available a lot of artificial intelligence approaches to process your images and so on, but in some cases it is better to obtain some good images firstly, then try some artificial intelligence and machine learning approaches to get something from these images. Because again, for example, in case of poor staining, when you have some strange dyes, and in this case it's again a nanoclusters, and as on these pictures, you can see here it's a cell, but it's me who sees the cell, and what about some machine learning? Would you able to adjust your scripts to see the cell or not because of poor staining? It's like maybe a couple of endosomes inside. And this is a very good question. When you stain something, you need to observe your images and your samples very accurately, because you need to understand what you are really seeing. And in case you have some additions to your cells, for example, here is a phosphatidylhexane phosphate, so it's a gel, and your cells incubated with gel, and it's definitely some structure which will scatter the light, and not just light but fluorescent light too, and this will be some artifacts in your sample, some additional lines like here, and all of this will affect your final quality of image. It's again going back to first questions on the first slide in fluorescence. In fluorescence, I ask you what should be the quality of your sample. If you have some gel, if you have some other additions in your cells, it would be very hard to obtain some very well looking sample. So to obtain some images even like this one, very clear, every organelle can be observed, this is just a very low magnification, so everything is clearly seen and so on, and just compare with this one. Well, it's not a fluorescence, of course, it's just a phase contrast, but if we will switch to fluorescence, there will be like maybe a lot of scattering, a lot of artifacts and so on. So it's always better to make a very good sample than to try to process these images and conquer some problems and so on. But there's also can be some other cases. For example, in high concentration of silver nanoparticles, a couple of times I observed these results. There was some false staining, false fluorescence as I call it, because this image was obtained in not in even fluorescence. In fluorescence, it's just an optical image with just a normal light. So there was so many of silver nanoparticles that I don't know how, but they maybe filter light, maybe they just interact with light in some other manner, because there's some plasmon properties of argentum particles. But in this case, there's no staining of cells, and even in the and still we see some red color. But there's no fluorescent, no propydium iodide, nothing. So this cannot be explained by just some common explanations. And so in some cases there can be ununderstandable results completely, and you should be aware of this, because these cells were stained with acridine orange. And as I mentioned, acridine orange can stain cells in green or orange, since it's acridine orange after all. Maybe there should be some, not orange, but red color, or maybe a bit yellowish. but still, as you can see here, it's well, 50 shades of yellow, 50 shades of green, and even some blue color. But there was no blue staining here, no blue dye here, and you can see a pure red, you can see green, you can see reddish, you can see, I don't know, in white some things. And these pictures were obtained maybe 5 years ago, and to this moment I don't understand how this happens. So sometimes you need just to redo the samples and do not try to explain, because sometimes it just cannot be explained. It's just something happened. Maybe probably because the cell were dying, and in some cells, as you can see here, it's a lot of organelles which stained inside, a lot of these small spheres, these apoptotic bodies. So these cells undergo apoptosis, and maybe because of different pH inside of these apoptotic bodies, acadine orange show a completely different color, so like 50 shades of orange. So what about DNA damage? Again, there's some options to stain DNA damage, and of course some DNA damage connected with reactive oxygen species and so on. But we talked a lot about how to stain DNA. Even acadine orange can stain DNA in red or green. But there are also some special staining to observe DNA damage or other options related to cell division, for example, cell mitosis. So one of the mostly used, it's Bordeaux staining, which is brom deoxyuridine, and this is the artificial analog for thymidine base. So during the mitosis, if you stain your cells with Bordeaux, you will observe some staining inside the dividing cells. So when the cells dividing, this stain can stain the dividing cells, and for example here you will see it's a cryozotanib, it's anti-cancer drug. Tinib, it's well, it's a lot of anti-cancer drugs which have this tinib ending in the designation, and in control samples you see some of the cells dividing, so showing in green. But at high concentration of anti-cancer drug, well, definitely cancer cells will die. No cells will not divide, and as you can see, there's no cells in mitosis. So the drug is working, the staining is working too. And this is just an example of counting of the cells, maybe on fluorescent microscope. But you also can use the staining in some histological examination. For example, maybe it's a kidney tubulus. I showed you already these histology patterns. And as you can see, it's some cells which divide in, probably it's some tubule epithelial cells, kidney epithelial cells. And as you can see, they are like inside of tubules, so it's just a kidney epithelium. But just to note, not all of the epithelium dividing. So I don't know. I do not remember exactly what this sample is, but still you can use it even in histology to observe some fossil dividing cells. And so this is a slide you already see about the comparison between overall viability and DNA damage. So before we discussed direct ways to observe viability, yes. So the staining, you can observe what happened to your cells directly with your eyes. So if you stain your cells with Bordeaux and you see there's no staining, it's probably something happened to mitosis at mitosis stage. The same in, for example, acadine orange. If you see that the nucleus of your cells stained only with acadine orange nucleus are red, it means that DNA is undergoes some damage. So DNA should be stained in green, and the nucleus of living cells should be stained in green. If the nucleus is stained is red, there is damage to nucleus, it's single-stranded DNA, and probably the cells undergo apoptosis. So, but again, this is direct way. If you observe with your own eyes, well, indirect. But in case of some metabolic activity test, as here it's probably MTT test and LDH test, you see there's no damage to cells, for cell viability like 100%, maybe a bit decreasing, but according to additional staining, you see that there's some, especially this one, it's a product of, as I mentioned, product of DNA oxidation. You see it's a lot of this product inside nanocobalt sample, and like maybe twice increase, and there's no twice increase at this concentration of 15. So neither here, neither here. But what else was done in this paper? Because there one additional very frequently used test to assess damage to DNA called comet assay test. Why it's called comet assay? Because the result of this test looks like a comet. And again, what we see here, we see that according to HOHDG staining, there's a lot of oxidized products after interaction with nanocobalt particles, increased twice compared to control samples, so titanium dioxide particles. And comet assay test, the basic principles is that you stain your DNA in your cells, and then you put your cells in agarose gel, and well, like a gel electrophoresis, but for a whole cell. And if you have some damaged DNA, this means that this DNA have lower molecular weight because it's damaged, like cleaved or something like this one. And this DNA will most likely exit, maybe not just exit the cells, but will exit the cells following the gradient of your electrophoresis. So in case of control cells, there is no comet tail, so they are called tails. In case of antioxidant molecules, they're not very long tails, but still. But in case of nanocobalt particles, you see a very big tail. So this tail not even twice bigger. If you're looking on this picture, not even twice, maybe, I don't know, well, maybe twice bigger. But in control, it's like a zero tail. When you have some catalase, it's antioxidant molecule. Their tails decreasing, and acetylcysteine, it's also antioxidant molecule. We see that the tails also decreasing, so the antioxidant molecules works, and it's only from this picture you can already give some conclusions that these particles, nanocobalt particles, have some additional generation of reactive oxygen species, and this species damage your DNA, and addition of antioxidant molecules can reduce damage done by these particles. So just from one picture you can produce all of the conclusions. But there's also other types of staining. For example, this one, again, very specific staining called H2X, and this staining use to observe damage done to histone groups. So as you know, DNA is wrapped around histones when they're in chromosomes, and these histones, there's a lot of them, it's a lot of families, and one of the histone families, H for histones, one of the families, H2X, and well, since there is a protein, you can use some antibodies to stain these histones and observe some damage done to these histones, because damage to these histones is associated with DNA damage, because it is known that some of phosphorylation of these histones is important when cell need to repair damage done to DNA. So again, it's the same paper as here. They performed a lot of experiments, as you can see, cell viability, different staining, and so on, even specific immunofluorescence staining. And well, after incubation with nanocobalt particles, you can see here it's staining with antibodies, and it's a lot of, well, looking like a violet, but it's just the interference between red and blue, so it's a lot of dots showing damage done to H2X, and there's almost no damage done by titanium dioxide. So it's a lot of different options to stain damage done to DNA. Most of them are expensive, but you can even use some Acadin orange to observe damage done by nanoparticles. And all of this is direct ways to assess viability, because if you can see that there's some damage done to H2X, it's definitely some damage done to DNA, and probably the reason for the reason of death of your cells, it's damage done to DNA. But there's also some indirect ways to observe cell viability, and it's called metabolic assays or colorimetric assays. So why it's indirect way? There's some types of metabolic assays which is very frequently used in our lab, in other labs, and so on. Maybe the most frequent one is the MTT assay, but it's also in some papers you can see LDH assay or LDH test, or OZ blue, LMR blue, Resurian test, and so on. What is the principle of all of these tests? So all of them are based on almost the same principle, that you have some molecule, let's say, and this molecule can undergo some transformation to another molecule upon interaction with mitochondrial oxidoreductases or some other molecules, some other enzymes inside the cell. So in case of LDH test, it a bit different from other tests, because it measures amount of LDH, lactate dehydrogenase, in cell media. So if your cell is damaged, LDH going out of the cell. Lactate. So how this LDH is produced? It's produced during the course of lactate to pyruvate cycle, and upon interaction, so this LDH can further well, not LDH, but this kit is just measured LDH released in in culture media, so you don't need cells for this kit to work. And there's also some formazan compounds like in the MTT, because formazan is very well studied and so on. Well, after interaction, after oxidation of NADH, you will absorb some electrons additional, which can produce some colorable compound, as well as an MTT assay. But MTT assay is an assay which assessing some work of mitochondria. So MTT assay, it's a very long compound, like you can see here, and this compound is yellow, but inside a cell during oxidation it can go transfer to water insoluble compound with some violet color, and this compound can be dissolved in DMSO, and you can observe some absorbance on this wavelengths. There are some additions to MTT family, not just MTT but TT family, let's say, because it's XTT, VST, and so on. And the main difference between all of the assay of this family is that some of these assays are water soluble, and some of them are more, let's say, precise, more accurate when counting the cell amount, because, for example, for MTT assay, there should be no less than 10,000 cells in your culture well, in each well. But for example, XTT assay can detect the viability of maybe 1,000 cells. So 10 times more precise when we're talking about results. But all of these tests, they mean that you need to destroy your cells to observe something, to observe some results, obtain some results. But in case of resurgent test, you can again, the principle is mitochondrial dehydrogenasis, and but after incubation with resurgent, you can further incubate your cell in your culture media. So this test is non-toxic, let's say. Because here you need to dissolve your cell completely, and here you need also to destroy your cell in ice, so homogenize them. And this is very useful test when you don't when you have not much cells, or you need to add something to cells to incubate them and so on. So you can stain your cells with this resurgent maybe multiple times. But again, why these assays are indirect ways to observe cell viability? Because they give you no information why the cell viability decreasing. So the decrease in cell viability means that there is decrease in mitochondrial oxidoreductases. So it's even opposite. A decrease in mitochondrial activity will tell you that there is decrease in viability. In other words, this I don't know if this term is used in English because it's in some cases it's called not even metabolic activity, it's called a breathing activity, because for cells, mitochondria, it's like lungs, and the cell need to breathe to produce some ATP and some energy, and but the reason for decreasing of mitochondrial activity cannot be obtained during these tests. So you cannot understand what happened to your cells. You just can say that something is happened. But to understand what precisely happened, you need some maybe fluorescent staining, some flow cytometry, some confocal studies, and so on. And that's why it's good, it's a powerful instrument and so on, but you cannot obtain a lot of information from this one. And there can be also even some misunderstandings when you're using some of these metabolic assays, some colorimetric assays, call them as you want, because just look at this picture, at this histogram. In some of the previous tests, you already see questions. Question 1 about anti-cancer drug used for glioblastoma treatment in brain, and this drug can kill the cells only during mitosis. So this drug is called docetaxel. And this drug is, interacts with tubulin structures, and so the cells cannot complete mitosis. So they enter mitosis, but cannot complete and dying. That's why for this drug, it's very important for this drug to interact with cells during mitosis. So what we see here, it's a very long, very big range of concentration studied, but forgive me, I just do not translate this, but it's hours. So 24 hours, 48 hours, and 72 hours. And as you can see, there's almost no dependence from concentration. The concentration changes from its nanomoles to micromoles. So the difference is 20 times maybe difference between these two concentrations. And as you can see, the difference in metabolic activity, like maybe 70% here and like 50% here, only 20%, but increase was 20 times. And as you can see, the second day again, there was a decrease, and the third day again, there was a decrease. But this decrease, it's concentration independent. And that's obvious to ask why. it's concentration independent. So in most of the cases, in most of the anti-cancer drugs, there should be zero at this concentration. If there is even nanomoles concentration reduce cell viability to 20%, 20 times more concentration definitely should go to, well, it's almost a zero, but still it's not like, I don't know, it should looks like this one, maybe just some exponential decrease. And when you are trying to add some let's say additional experiments like mix, this is the MTT assay. And when you use some fluorescent staining to understand what is going on, well, in some cases you can obtain this answer, but you need to think a bit to understand what really happened to your cells. Because here what we see, 24 hours, it's a concentration of almost 2 micromoles. As you can see here, it's on one of the biggest concentration used. And according to MTT assay, after 24 hours, the concentration of dead cells is like 43%. As you can see here, it's well, maybe it's just another cells, but it doesn't matter. Okay, there was a couple of these slides. Maybe I take a wrong slide. It doesn't matter. So according to MTT assay, it's like a 43% of dead cells, but according to fluorescent staining with NXene, it's like only 50% of cells undergo, well, dead, so they have a red nucleus. And the difference is three times. And after 72 hours, staining with NXene gives like 55% of the dead cells, but the MTT gives like 90% of the cells are dead. So it's just not dead, it's just a decrease in viability. So it's either can be 90% of cells are dead and 10 survived, or every cells are survived, but they are living only just for 10% each. So you can't even tell what is happened to your cells. And the most important, why so big difference? Here is 3 times, here it's more than 30%. So what happened? How this can be explained? Because you need to give some, tell some conclusions about is it a good drug or not good drug, can it be used as anti-cancer drug or not? And the answer is, well, again, if you remember at the start of the lecture, when you have some reactive oxygen species or some damage, it doesn't matter what is here, reactive oxygen species or anti-cancer drug or something like this. When something happened to your cells, especially to mitochondria, it will definitely cause a decrease in ATP. And ATP is the most important molecule when we are talking about initiation of regulated cell death. And as you can see, these levels of ATP can decrease dramatically, maybe 3 times, or maybe even become even lower. And there's still some possibility for the cell to survive even here. So there should be some point of no return. And what happened here? Here you see some decrease. So MTT, it's measuring, well, in other words, it's measuring ATP concentration. And according to MTT, the decrease is very, very high. But according to direct observation with fluorescent staining, there's no much cells dead, because these dead cells, like 15%, they are here. So some of the cells already passed this point of no return, but most of the cells are still on the way to this point. And the same is here. After 3 days, more and more cells dying, and almost all of the cells are dead according to this one, because almost all close to this point of no return. And well, according to NXene, some of the cells passed this border. And why this takes again, but the question is why this looks like stages, like as you can see, it's almost a horizontal stage. Maybe some decline, maybe just a very slow decline, because this drug working only during mitosis. And when you have, and in this particular example, all of the cells was in G0 or G1 phase. So this was a synchronized cell culture, and there was no mitosis in this culture. So this doesn't matter how many drug you put in your cells. If no cells undergo mitosis, there will nothing happen to them, because it's working only during mitosis. Of course, they uptake a lot of drugs inside, but it doesn't matter because it's not working until they undergo division. And this division occurs simultaneously in some cells because, well, some cells dying from maybe other reasons. And because this culture is synchronized, they dying almost simultaneously, and this is looking like a stages. But the question is that when we are talking about some nanoparticles, what was the case of this drug? There was a case that we need to enhance enhance the anti-cancer properties of this drug by using targeted delivery principles. And since we need to enhance this drug, we need to understand which concentration we need to enhance. And this is very important because, as you can see here, all of this concentration works quite good. Well, 20% or 80% decrease, so it's a very, very high. But we need to find the concentration which not working. And here again you can see some even more precise. Well, it's not a micromole, it's a nanomole should be here. So it's a dissolution of drug from this point down to nanomole, and this is not even a nanomole, it's already picomole concentration. And you can observe here that at some concentration, finally, even after 72 hours, there is no cytotoxicity observed. And what we aim to find is here, it's some, well, a bit exponentially looking decrease in cell viability. And this is a concentration we need to enhance, because this is already working very good, this is not working at all, and this is some concentration. In case we enhance this enhance cytotoxicity of this concentration, we can enhance the effectiveness of this drug. But when we're talking about the cells, it's not in some ways obvious, because this is a very low concentration. If you calculate amount of molecules of docetaxel cell, thinking that this is not a micromole, it's 24 nanomoles, and in each culture well, we have like 100,000 of cells, a lot of cells. And if you divide amount of molecules divide on amount of cells, there will be some countable amount of molecules per each cell. So this drug is very toxic, very, very toxic, and its use limited to stage 3 of breast cancer, of brain tumors like glioblastoma and so on, because it's a last line of anti-cancer therapy, because it's a very, very toxic drug. And to enhance the ability of this drug to kill the cells, this concentration is probably some kind of promising. But when we add some targeted moiety using gold nanoparticles, and we deliver some of this docetaxel drug in our cells, there was completely opposite results, because as you can see in these circles, a blue bar is higher than orange bar. And what's so special, you ask? Because Blue bar, it's a dot-stack cell conjugated with nanoparticles, and as you can see, cell viability increasing after conjugation. So we add some targeted delivery moiety, and this targeted delivery moiety made things only worse. So in some cases it's even higher than 100% viability. So we enhance proliferation of cancer cells. Again, scientists discovered some weapon, tumor genesis weapon. What really happened, and also here you also can see that there's something bad happened. So what's the answer to this one? Well, in some cases, why I'm telling you all of this? Because this is the results of MTT test. And obtaining these results, you are not able to say what happened to your cells. Why? You add some anti-cancer drug. You conjugated your anti-cancer drug with some nanoparticles. These nanoparticles can be toxic, probably. This drug should be better delivered in these particles, accumulate in these particles and cause some additional cytotoxicity. But according to MTT assay, everything is bad. Everything is worse than in case of just pure drug. And these results cannot be explained by only data obtained from MTT assay. So what was the answer? The answer was very simple. Now, as I mentioned, the countable amount of molecules per each cells, and in case of absence of nanoparticles, these molecules spread in cells, well, kind of uniformly. So each cell receive like maybe 100 of molecules of this dot-stack cell drug. But in case of conjugation with nanoparticles, the concentration remains the same, but since this concentration is very, very low, it's a micromole concentration, so in case of conjugation, some of the nanoparticles received a lot of molecules on their surface, and some of the nanoparticles not. So in case nanoparticle enters the cell, and on the surface of these nanoparticles, it's like 1,000 of molecules of this dot-stack cell drug. And for the cell, it's even 100 enough. There is no need again in 1,000 of molecules. So this cell will definitely die in next phase of mitosis. But on the surface of some nanoparticles, there is no molecules because the concentration is so low. And because of this, some of the cells receive nanoparticles without no drug, and these cells will survive. And because of the survival, survival rate, we see some increase in viability of the cells. Because for such a low concentration, targeted delivery approach may not work, because concentration very low, and amount of particles enter the cells also is very low, and some of the cells receive a lot of drug, some of the cells receive no drug. And you should perform some theoretical calculation to understand this, because you will not able to obtain this information directly from MTT assays. And again, what are the problems? This is probably last thing I need to discuss today. What other metabolic assays related problems can be obtained without additional fluorescent staining and microscopic observation and so on? Again, it's from my practice, but it's a good example of what can happen to the cells upon interaction with two toxicants. Here is silver nanoparticles, which is very toxic, and this is a surfactant stub, which is also very toxic, and they are added to the cells in mixture. So this is not stub stabilized particles, it's just stub plus nanoparticles. And as you can see, at high concentration, we see some decrease in viability by 70%. But in case of lowest concentration, here and here, we see an increase in activity of the cells. By the way, it's just epithelial cells, not a cancer one. It's just a normal cells. So we see increase in activity of these cells, and this is 24 hours, so 1 day passed. And after the second day, we see even a bigger decrease in activity. So what happened? We add and we see even some increase in activity in the highest concentration. So how it can be possible? It just cannot be possible at all that we have added two toxicants and that cytotoxicity decreases. So not even decreases. We have some proliferation, additional proliferation up to 40%. And this is 48 hours. And after 72 hours, finally, the cells died. And what is the answer to this phenomena? The answer is in the picture, again this one. As you can see, when ATP and ROS concentration changes, there can be some adaptive response period with some fluctuation of concentration of ROS and ATP. And since there's a fluctuation in ROS, what is the cause of this fluctuation? Because, well, the cell start to produce antioxidant molecules, basically glutathione dehydrogenases and catalases and so on, to combat this reactive oxygen species. And one of the main toxicants, cytotoxicity, cytotoxicity associated damage done by silver nanoparticles, it's a ROS generation. So here we have some ROS generation, and cells activate their capabilities and start to generate some additional molecules. And it's like a human under stress, under some stress condition, when the human can lift something heavy, can jump higher, run faster, and so on. So the cells activate some internal capabilities to withstand the damage done by these particles. And this was on the first day, on the second day, and finally capabilities ended. So there are some, let's say, some storage of these molecules, some abilities of the cells to withstand, but these abilities are limited. And on the third day, these abilities just ended and the cells died. And this can be easily understood, can be easily revealed if you stain your cells for glutathione concentration, and you will observe a decrease on the third day. So using just a metabolic activity test like MTT assay test, you will obtain these results, and these results will look for you very strange, but to understand what's really happened to your cell, you will always need some direct ways. So you need to observe your cells with your own eyes and understand what happens. And to use some fluorescent staining, fluorescent microscopy to understand what happened to DNA, what happened maybe to reactive oxygen species level in your cells, to add some dCFDA in your cells and so on. So always MTT assay and all other assay, it's indirect ways, and they give you just a basic information which cannot be further used. Just only MTT. It's not the best option. And also there can be some problems when you test nanoparticles in a very high concentration, like this one. It's 200 milligrams per liter of argentum particles, and you can see it's a control well, and this is a well with nanoparticles, and they will be definitely and always and also here, if you even observe your cells directly with your own eyes, it's a lot of interference from this optical interference of these nanoparticles with your MTT assay. So you need to carefully conduct, modify your protocols for MTT assay to assess viability of the cells when we are talking about so high concentration. And as you can see here, it's even more higher, twice higher concentration here. So you need to adjust your protocols to meet this criteria of that high concentration. Well, that was the last slide. There's a lot of information today, and I just hope this was useful for you. And if you have any questions, you are free to ask again in chat. And maybe in 10 minutes, 5 minutes, 10 minutes, I'm waiting for you in Zoom room, because some of you should present the topics. If some of you are not able to present today, the last chance will be during consultation after the last lecture in Friday. So here's the last chance, today or Friday. I see no questions probably. Okay, no questions. See you in Zoom room. Link here. Schedule in your common folder. And see you on last lecture. Some of you in 10 minutes, and some of you on last lecture this Friday.